briefly take questions?
Yeah, if you're running behind, yeah. Okay, sure.
Ladies and gentlemen, I'm sure you'll agree that that was the most inspiring lecture, made very, very simple for all of us to understand. Professor Berkovic will take some questions. Please do introduce yourself and ask your question. I would request you to be brief and keep it simple. Thank you.
Dr. Veeravalli Raghavan, another Indian name for you. You see, you mentioned the British Raj. There was a good British doctor who said, in the tropics, if you encounter philosophy, suspect malaria. Now, you see, I don't know how many people understand that, but I'm afraid your attitude, when you say that you can dismiss mysticism, I asked you this question before the lecture, are you going to talk about self-transcendence? And you said no. That seems dismissive to me, because Rama's book, The Phantoms of the Brain, chapter 9, I think, and Oliver Sacks' book, Hallucinations, chapter 8, they both talk about self-transcendence. The route to self-transcendence may lie through epilepsy. So the sacred disease cannot be dismissed so easily. That's my feeling.
Thank you for that comment, sir.
Yes. Karunanidhi, I'm a diabetologist, but I train in our Institute of Neurology as well. That's thanks to the synapse of Srinivasan and synapse of Srinivasan between VHS and Venu, we could hear your Melbourne melody, sir. The question is that, as you rightly said, the precision medicine, all WES, all exams, genome is becoming, as the technology matures, WES is becoming a test of choice. It would help us not only to know about the nature of the etiology of the seizure, but also the response to the medications. Do you think that we in the developing countries would be able to harness the technology? Because here, the out-of-pocket expense, as the Stanford speaker said about the expense of seizure medications.
Thank you.
Yeah, so it's a very good question. Is this technology going to be applicable to developing countries? I hope so. I mean, it wasn't applicable to the developed world until a few years ago, and as I've said, the cost has come down from 100 million to 1,000, and there's actually now a new wave of technology where it's probably going to come down more, and there are projections that maybe it can be done for $100 in the not too distant future. Now, that may be even too much for some areas, but it is, the changes in technology are just spectacular. So I'm hopeful that it will be more widely applied. And you're right, I did not mention response to treatment predicted by genes, so-called pharmacogenetics. That is a very important feature. This work has to be done with very large collaborations. So we're working with groups all around the world. In fact, we had discussions just 2 days ago about including potentially a large number of Indian patients in a research project to sequence. We have a project called 25K where we're planning to sequence 25,000 patients with epilepsy that's been funded by the NIH, and that's a worldwide collection. And we very much want to include all ethnicities and races.
Mr. Ram.
Professor Berkovic.
Oh, sorry.
With these revolutionary advances in tackling the problem, do you see any prospect for preventive approaches apart from
look, that's a very good question. There is certainly evidence if you detect lesions early and remove them that they help. There's some very interesting data in a condition called tuberous sclerosis where patients develop, it's a genetic condition, so you know that the child's going to get it. If they're treated early, that can help. And we're hopeful that application of these precision therapies very early on might do that, but we don't have proof of that. But I agree with you, that would be a wonderful thing.
Professor Kamakshi.
I'm Dr. Kamakshi Shanbog, formerly HUD Institute of Neurology, long time back. My question is, sir, there is a condition called genic pleomorphism and genic pleiotropism. That is a single gene producing multifarious manifestations. Maybe in the father it may be one type of epilepsy and in the offspring another type. Can this theory hold good for any differences in the epileptic patterns of parent and offspring?
Yeah, so it's a very good question. There is variation seen within families where they have the same major genetic change. We don't understand why that happens. It's something we're very, very interested in. We think it's due to so-called modifying genes, that even people in the same family have a different genetic makeup. Parents and children share half their genes. So we think it may be the other genes interplaying, but that's an extremely difficult thing to measure, so we don't yet have the answer.
Thank you, sir.
Yes.
I'm Adarsh, a third year MBBS student here, a medical school student. If epilepsy is associated with some genetic defects, can it be cured or reversed by some genome editing techniques such as CRISPR? If so, do you know any studies which are doing it?
Okay, so the question is whether you can actually fix the gene or gene therapy. A few years ago, we would have said absolutely not. And in fact, attempts at gene therapy in the late 90s were actually disastrous, and it actually put the field back many years. There has been just now a remarkable study published not on epilepsy, but on a condition called spinal muscular atrophy, where children get weakness of the muscles due to degeneration of the nerves, which comes from the spinal cord. And they've been able to improve that by injections of the spinal cord of what are called antisense oligonucleotides. These actually latch on to the abnormal gene and reverse the change. And the findings are persuasive, although it's not yet absolutely definitive. And this sort of approach might well be applicable to the epilepsies in the future. It's not there yet, but we're hopeful that it may be applicable. It's still science fiction, but maybe not that far away.
Are there any studies carried out right now? Do you know of any?
I don't know of any study. There are studies in animals with animal models, but not yet in humans. That's a big step to do this in human patients, particularly children. We have to get a lot more data to be sure that it's likely to work and likely to be safe.
Thank you, Professor Berkovic. I'm sure we can keep asking questions, and we can continue that in high tea that will follow. It's my pleasant duty to say thanks. I'd like to begin by saying a very big thanks to Professor Berkovic and Mrs. Berkovic for coming all the way from Melbourne and making a very complex topic very, very simple for all of us to try and understand. A big thanks to Professor Pedley and Professor Matthias for being here with us today. We're very, very happy to welcome both of you back. To Professor Satish Chandra from NIMHANS for all his support, and Professor Gangadhar in absentia, because they've both been very responsible for the program in NIMHANS. Very grateful to both of them. The entire Srinivasan family for years and years of support and making this such a wonderful program that all of us are able to enjoy each year. In particular, I'd like to thank Mrs. Prema Srinivasan, who's here with us in the front row today. Thank you very much, Auntie. Like to say thank you to the press for being here in large numbers, to the various people who are involved in organizing this, both from the TVS group and otherwise, and to all of you for taking time off on a Sunday to come and listen to this lecture. Please do join us for high tea. Thank you so much.
Schooling, but I enjoyed it. I learned more.
He's got a very perceptive mind, your son.
saying that because we had a chance to talk.
It is doing so well, and I said, you will just patient problems go home quickly, and I could not see hospital. Father will be time if they come avoid.
You're his aunt. Okay. Well, when I come across, I don't tend to come across the procedures all that often. Just want to recommend. Okay.
Doing it anymore. Doing it anymore. They will and immediately once it